Greetings everyone, I'm Maria Cristina delasllagas, a professor of Ateneo de Davao University in the Philippines. In this lecture, we are going to talk about the overview of transcription in prokaryotes and eukaryotic cells. Much of the progress in biology has happened over the past century. In the previous lecture, we learned about Mendel's work that genes are discrete elements that govern the appearance of specific traits. The study of Boveri, Weismann, Sutton, and their contemporaries discovered that genes have a physical embodiment as part of the chromosomes. We learned from Morgan and colleagues that genes have specific addresses, meaning they reside at particular locations on a particular chromosome. The study of Griffith, Avery, Hershey, and Chase demonstrated that genes were composed of DNA, and in the previous lecture, we learned that Watson and Crick solved the puzzle of the DNA structure. Although these different concepts were milestones along the path to genetic understanding, it did not explain the mechanism by which the information stored in a gene is put to work in governing cellular activities. In today's lecture, this will be our major focus by talking about the relationship between genes and the polypeptides, describing the flow of information through cells, and we will try to elaborate on the differences between transcription in prokaryotes and eukaryotes. One of the most important insights on the relationship between genes and proteins was provided by the observation of Dr. Archibald Garrod, a Scottish physician, who reported in 1908 that the symptoms exhibited by persons with certain rare inherited diseases were caused by the absence of a specific enzymes. One of the diseases that Dr. Garrod investigated was alkaptonuria. This is a condition characterized by the presence of a dark urine after it is exposed to air. Dr. Garrod found that persons with alkaptonuria do not have an enzyme in their blood that can oxidize homogentisic acid, which is a compound formed during the breakdown of the amino acids phenylalanine and tyrosine. Because this homogentisic acid accumulates, it is excreted in the urine and it darkens in color whenever oxidized by air. So from here, Dr. Garrod discovered the relationship between a genetic defect, a specific enzyme, and a specific metabolic condition. He called such diseases as inborn errors of metabolism. Well, of course, this observation of Dr. Garrod, while we find it important now, is unappreciated for several decades. Another early insight on the role of genes in the direct production of enzymes worth mentioning was by the study of George Beadle and Edward Tatum of the California Institute of Technology in 1940s. So they studied Neurospora, which was a tropical bread mold that can grow in very simple medium containing a single organic carbon source, inorganic salts, and biotin. So because Neurospora needs very little to live on, they were thinking that Neurospora can synthesize all of its required metabolites. Therefore, the organism would be very sensitive to enzymatic deficiencies, and they can easily demonstrate this through experimental protocols like what you see here on screen. Beadle and Tatum's plan was to irradiate mold spores and then screen them for mutations that cause the cell to lack a particular enzyme. In this experiment, Neurospora was exposed or irradiated using X-ray or ultraviolet light in order to induce mutations. And then, it was allowed to grow into colonies in tubes containing supplemented medium. Genetically identical spores produced by the colonies were then tested for their ability to grow on either supplemented medium or minimal medium. Those that failed to grow on minimal medium were the mutants. And so the next step is to determine the mutant gene. In this example, a sample of the mutant cells was found to grow in the minimal medium containing vitamins, but it would not grow in medium supplemented with amino acids. This observation only indicates that there is a deficiency in an enzyme leading to the formation of a vitamin. And so to find out what gene is mutated, you could grow the mutant cells in minimal medium supplemented with different types of vitamins to find out which enzyme is deficient. For example, if the cells are capable of growing in a medium supplemented with pantothenic acid, it only means that there is an enzymatic deficiency that prevents pantothenic acid production. On the other hand, if the mutant will grow in a minimal medium supplemented with riboflavin, it means that there is an enzymatic deficiency that prevents the production of riboflavin. In eukaryotic cells, the DNA of the chromosome are found within the nucleus. Information present in a segment of the DNA is made available to the cell through the formation of a messenger RNA molecule. And the process by which the messenger RNA molecule is formed is called transcription. The word transcription tells us that the information encoded in the DNA is transcribed or is rewritten into a similar language in the form of a messenger RNA. Messenger RNA contains complementary information to that of the gene. And because of this, the messenger RNA can also be described as a sense strand or coding RNA. It is important to note that eukaryotic messenger RNA are not synthesized in their final or matured form, so it's called pre-messenger RNA, because there is still a need to process it in order to form the matured messenger RNA. So the matured messenger RNA will be used in the cytoplasm, whereby the information the mRNA carries will then be translated into proteins. Messenger RNAs are very important, so it allows the cell to separate information storage, which happens in the nucleus, from information utilization, which happens here in the cytoplasm. So while the gene remains stored safe inside the nucleus, information from the gene is being used in the cytoplasm in order to form functional proteins. So the use of the messenger RNA therefore allows the cell to amplify its output, which happens to be the protein, because one DNA molecule can serve as a template to form a lot of messenger RNA molecules, so that there could be more polypeptides produced in the process. Proteins are synthesized in the cytoplasm by a complex process called translation, and translation requires not only messenger RNA molecule, but other classes of RNA molecules. For example, the ribosomes contains both protein and ribosomal RNA. So ribosomal RNAs or rRNAs are essential as a structural component of the ribosome as well as for catalyzing the chemical reaction in which amino acids are covalently linked to one another. Transfer RNAs or tRNAs is another class of RNA molecule that is required during protein synthesis. The transfer RNA are necessary to translate the information in the messenger RNA nucleotide code into the amino acid of a polypeptide. Note that ribosomal RNAs and transfer RNAs owe their activity to their complex secondary and tertiary structures that are formed as a result of RNA folding. And RNA folding is driven by the formation of regions having complementary base pairs. For example, in this figure, you can see the different regions of the single strand that base pair with each other. The expanded portion here, this one, expanded portion, shows the base sequence of a stem, this is the stem, and the loop. It also includes the non-standard base pair, guanine and cytosine here, as well as a modified nucleotide, this one here in light blue, the methyl adenosine. Transcription is a process in which a DNA strand provides the information for the synthesis of an RNA strand. The enzymes responsible for transcription in both prokaryotic and eukaryotic cells are called DNA-dependent RNA polymerases, or simply RNA polymerases. These enzymes are capable of incorporating nucleotides one at a time into a strand of RNA whose sequence is complementary to one of the DNA strand, which serves as a template. The first step in the synthesis of RNA is the association of the polymerase with the DNA template. And the site on the DNA to which the RNA polymerase molecule binds prior to initiation of transcription is called the promoter. Cellular RNA polymerases are not capable of recognizing promoters on their own. Instead, they need the help of additional proteins which we call as transcription factors. In addition to providing a binding site for the polymerase, the promoter contains information that determines which of the two DNA strands is transcribed, as well as the site at which transcription begins. In this diagram here, the elongation of a newly synthesized RNA molecule during transcription is shown. The polymerase covers approximately 35 base pairs of DNA. Meanwhile, the transcription bubble created here is composed of single-stranded or melted DNA having around 15 base pairs. And the segment present in the DNA and RNA hybrid here would include around 9 base pairs. The enzyme would generate overwound or positively supercoiled DNA ahead of the transcription site, and it also creates an underwound or negatively supercoiled DNA behind itself. Now, these conditions are relieved by the help of the enzyme topoisomerase. In this diagram here, the RNA polymerase is in the act of transcription elongation, whereby the downstream DNA lies in a groove within the polymerase here, and it is clamped by a pair of jaws formed by two large subunits of the enzyme. The DNA makes a sharp turn in the region of the active site so that the upstream DNA extends upward as shown in this diagram. Meanwhile, the nascent RNA here exits the enzyme's active site through a separate channel shown in yellow. RNA polymerase moves along the DNA template strand towards its 5' end, as you can see here. As the polymerase progresses, the DNA is shown to be temporarily unwound, and the polymerase assembles a complementary strand of RNA molecules, which grows from a 5' to 3' direction. So there. The nucleotides that enter the polymerization reaction begins as a ribonucleoside triphosphate substrate, and eventually these are cleaved into nucleoside monophosphate in the presence of the enzyme. And this enzyme is the pyrophosphatase, which allows for the hydrolysis of the pyrophosphate, converting it into inorganic phosphate. The energy released in the reaction allows for the elongation of the growing RNA molecule. Once the polymerase has finished adding nucleotides, the DNA-RNA hybrid will dissociate and the DNA double helix will reform. It must be noted, however, that a single DNA template may be transcribed by several RNA polymerases so that it can produce a lot of messenger RNA molecules. Transcription in bacteria like the E. coli contain a single type of RNA polymerase that is composed of 5 units that are tightly associated to form a core enzyme. If the core enzyme is purified from bacterial cells and is added to a solution of bacterial DNA here, and ribonucleoside triphosphates, the enzyme will bind to the DNA and it will synthesize RNA. However, the RNA molecule produced from the purified polymerase is not the same as that you will find within the cells because the core enzyme will attach to random sites in the DNA molecule. If, however, purified sigma factor here in purple is added to the RNA polymerase before it will attach to the DNA molecule, attachment of sigma factor to the core enzyme will increase the enzyme's affinity for the promoter site in the DNA, and it will also decrease its affinity for DNA in general. In other words, the presence of a sigma factor will allow the recognition of the polymerase recognizing the suitable promoter region in the DNA molecule. This is an X-ray crystallographic analysis of bacterial RNA polymerase showing a molecule that is shaped like a crab claw with a pair of mobile pinchers or jaws enclosing a positively charged internal channel. As the sigma factor interacts with the promoter, the jaws of the enzyme grips the downstream DNA duplex, which resides within the channel. The enzyme will then separate the two DNA strands in the region surrounding the start site. Strand separation makes the template strand accessible to the enzyme's active site, which you will see here in this area. Initiation of transcription will appear to be a difficult undertaking at first because the RNA polymerase will typically make several unsuccessful attempts in order to assemble an RNA transcript. And once around 10 nucleotides have been successfully incorporated into the growing transcript, like here, the enzyme will undergo a major change in conformation, and it will be transformed into a transcriptional elongation complex that can move along the DNA. In this model, the formation of an elongation complex is followed by the release of the sigma factor here as elongation occurs. Promoters are sites in the DNA that bind the RNA polymerase, and bacterial promoters are located in the region of the DNA strand just preceding the initiation site of RNA synthesis. The nucleotide at which transcription is initiated is denoted as positive 1, and the preceding nucleotide as negative 1. Portions of the DNA preceding or before the initiation site are referred to as upstream, while those that succeeds or follows the initiation site are referred to as downstream. Analysis of DNA sequences just upstream from a large number of bacterial genes reveal that there are two short stretches of DNA that are similar from one gene to another. One of these stretches are found 35 bases upstream, while another stretch is found 10 bases upstream from the initiation site. Now, these sequences are referred to as consensus sequence and generally consist of the sequence TTGACA. The consensus sequence indicates that it is the most common version of a conserved sequence, but that there are some variations from one gene to another. The second conserved sequence is known as the Pribno box after its discoverer, and it generally has the consensus sequence TAAT. The Pribno box is recognized by the sigma factor, and amino acid residues within the protein would interact with each of the 6 nucleotides of the TAAT sequence of the non-template strand. And 2 of these nucleotides are flipped out here. It's flipped out of the nucleotide stack and into the core of protein. And it's likely that this will initiate the melting of the adjoining region of the promoter DNA, and it will allow the formation of transcription bubble that we have seen earlier. Just as transcription is initiated at specific points in the chromosome, it also terminates when a specific nucleotide sequence is reached. In roughly half of the cases, a ring-shaped protein called the Rho factor is required for termination of bacterial transcription. The Rho encircles the newly synthesized RNA and moves along the strand in a 5' to 3' direction to the polymerase, where it will separate the RNA transcript from the DNA to which it is bound. In other cases, the polymerase stops transcription when it reaches a terminator sequence. The terminator sequences typically fold into a hairpin loop that causes the RNA polymerase to release the completed RNA chain without requiring any additional factors. Unlike prokaryotes that has only one RNA polymerase, eukaryotic cells have three distinct transcribing enzymes in their cell nuclei, and each of these enzymes are responsible for synthesizing a different group of RNA, as shown here in this table. Note that the messenger RNA are transcribed by RNA polymerase II, while the ribosomal RNAs are mostly transcribed by RNA polymerase I. In the meantime, the tRNAs are transcribed by RNA polymerase III. A major difference between transcription in prokaryotes and eukaryotes would be the requirement of a large variety of accessory proteins or transcription factors in eukaryotic cells. And these proteins play a role in virtually every aspect of the transcription process, from the binding of the polymerase to the DNA template, to the initiation of transcription, to its elongation and even termination. Now take note that all of the three major types of eukaryotic RNAs, the mRNAs, rRNAs, and tRNAs, all of them are derived from a precursor RNA molecule that is longer than the final RNA product. And this initial precursor RNA is equivalent in length to the full length of the DNA that is transcribed. And this is called the primary transcript or the pre-RNA. The corresponding segment of DNA from which the primary transcript is transcribed is called the transcription unit. To learn how eukaryotic ribosomal RNAs and tRNAs are synthesized and processed, please watch the next video. Another video will also cover the synthesis and processing of eukaryotic messenger RNAs.